Okay, I want to kind of look at chapter 17 with you. Chapter 17 is the chapter in the Hofnagel's text, and if you're looking at OpenStax, I think it's in the 20s. But we're looking here at two prokaryotic domains, archaea and bacteria. Pictures I have on the front here basically to indicate on the left-hand side that there are a lot of bacteria that are pathogenic that can cause disease, but there are also bacteria that are decomposers or important in your digestive system and so forth. So on the right-hand side, a picture from Yellowstone and some of the archaeans that live in these hot springs. And then probiotics, you've probably heard that term. But those are bacteria that are good for us, and a lot of them live in our digestive system and are helpful. So in terms of characteristics, prokaryotes are the smallest organisms that are considered living on our planet. When we say they're the most biologically successful, we're talking about the fact that they were there at the beginning and they're still around today, and they're very easily adaptable to the environment, and so they have lasted for a very, very long time. And so we have the two types, and they're actually in two different domains. So bacteria is one, it has its own domain, and then archaea is the second. So there are big enough differences in terms of some of their DNA and RNA and cell wall structure and things like that that scientists have decided that they each should get their own domain in terms of classification. So in your book you have pictures. The top picture is a compound microscope micrograph, kind of a different staining technique. The domain archaea picture is actually one that came from an electron microscope, and probably, well, it says false color, so somebody colored it. They don't really look at those colors. And then at the bottom is a tree that we will look at in this unit and the units to come quite a bit, just for overall big picture. So you notice that there is one stem on this tree, and that's basically where the common ancestor would be. And then you notice that bacteria take the left branch and archaeans take the right branch, and you notice that the archaeans are on the same branch as the domain eukarya, and so they actually are genetically and biochemically a little bit more closely related to us than the bacteria are. So we talked a little bit about classification in chapter 14, but they were named because somebody at the beginning thought that they must be the oldest living organisms, and now we realize that they're a little more closely related to us, so they're probably not the oldest. So I want to look a little bit at some characteristics and structure. So bacteria are pretty simple. I think we know that. They have only one chromosome, and it's a closed circle. So it's not the long straight chromosomes like we see in humans. And we don't call where this DNA is located a nucleus because there's no membrane around it, so it's called a nucleoid. So you can see that it's a pretty long piece of DNA. It's kind of jumbled up, but it's just one big circle. And then you can see that there are some smaller pieces of DNA in the cytoplasm, and those are called plasmids. And so bacteria are able to pick up DNA from different places, and so they kind of pile up DNA in their cells, which is one of the ways that they are able to adapt pretty well. Okay, they all have ribosomes because they need to be able to make protein. They all have a cell membrane, and then they have an outer cell wall that has peptidoglycan in it. So we're going to talk about peptidoglycan a little bit in lab, but just realize that this is a molecule that we only see in bacteria, and so it makes them different from any other organisms that have cell walls, and that's part of the way that we identify different kinds of bacteria is how the cell wall is organized. Okay, and then you do have bacteria that may have flagella or pili or different things, and I'm not going to worry so much about those extra parts, but just look at basic structure of the bacteria. So in your book you have a diagram of the cell wall structure for bacteria, and you notice on the left-hand side the peptidoglycan are the green structures in the image. And so it's a pretty thick cell wall on the outside of the cell membrane for one way of organizing the cell wall. And then on the right-hand side, you can see a really thin layer of peptidoglycan, which is the cell wall. But then in this bacteria, they have an extra membrane on the outside of the cell wall. So we'll talk a little bit more about that as we go into lab on bacteria. Okay, so a couple, the picture on the right, Vibrio cholerae, so that's the bacteria that causes cholera. You can see it has a flagellum. The picture on the left is basically just a generalized one where you can see all of the different things that might be found in a bacterial cell, but they don't all necessarily have, you know, the flagella and the pili on the outside and so forth, but gives some options for bacterial cells. Okay, so as a scientist, if you're studying bacteria, there are several things that we look at in terms of trying to figure out who they're related to, how to classify them. So we look at their shape, we look at how the cells are arranged, we look at the cell wall structure, we look at where do they get their energy, are they autotrophs, are they heterotrophs, and also aerobic versus anaerobic, do they require oxygen or can they live without oxygen? So we're going to look at examples of all of these criterion for classifying bacteria. So again, I'd just like to use some of the diagrams from your book, from your Hofnagel's book. So this is kind of nice because it indicates the similarities between bacteria and archaea, what they have in common, but it also shows you some of the unique characteristics that they have that differentiate them and put them into two different domains. You can see in the features unique, notice that the archaeans will never have chlorophyll, so they're not going to do photosynthesis. On the left, we do have bacteria that can have chlorophyll. You have the cell wall that has the peptidoglycan. So those are just, you know, you don't have to memorize all of those, but those are some things that separate them or differentiate them. Okay, so I'm going to throw in, hopefully in every discussion, just some interesting facts that you might think about in terms of whatever the subject is. So these two, we're talking about bacteria. So for perspective on how big they are, hopefully you can visualize how big a teaspoon is. Okay, so how many bacteria would you find in a teaspoon of agricultural soil? So I want you to think about that for a minute before we look at the answers. And then how many bacteria would you find in a teaspoon of yogurt, which is actually something that you eat, and it's considered a probiotic. Okay, so in those two, in agricultural soil, so soil where plants are growing, 1 billion bacteria in 1 teaspoon. In a teaspoon of yogurt, 20 million, still a lot, but of course there's in that yogurt there's also all of the organic molecules that make up the carbohydrates that are giving you nutrition from that yogurt. So first of all, in terms of characteristics, we're going to look at cell shape. Okay, and so some of these pictures that I'm using are electron micrographs, so they were taken by electron microscope. So they're real good for giving you a close-up look. The microscopes that we would have used in lab are compound light microscopes, so they don't have this much magnification. But the bacillus bacteria are rod-shaped, so you can see that they're kind of long and straight. You can see that we also have some, it shows that sometimes you can have them in a little bit of a chain, but each cell in that chain is a bacillus shape. Okay. We can have bacteria that are round, so coccus are round, and you can see in these two photographs that you can have individual round ones, or you can have round ones that live in a colony, and that's a chain, or it could take some other cellular arrangement, which we'll look at in a little bit. And then you can have the spiral-shaped ones, which are spirillum. So you should be able to identify a bacterial cell by its shape and give the correct terminology for the shape, scientific terminology. Okay, so this is what they would look like under a compound microscope, not as high magnification, but up in the top left-hand corner we have the spirillum. Down at the bottom we have the bacillus, and a lot of them are in a long chain. And then on the right-hand side we have the coccus, which are kind of grouped in little groupings rather than individual cells. Okay, so cellular arrangement I mentioned. So coccus means that it's round. Staph means that it's going to be in a grape-like cluster. So staphylococcus would be little round cells that you find in a cluster, like you see in the picture on the right. And then strep means that it's in a chain. So streptococcus, still little round cells, but they are grouped together in chains. So you've heard strep probably in relationship to strep throat, and so strep throat is caused by streptococcus bacteria. And then staph, you may have heard of also, staph infections. So MRSA, you do not have to be able to write out the whole what that stands for, but methicillin is an antibiotic, and so this is a methicillin resistant kind of staphylococcus, which means that it's very hard to treat and it's easy to spread. And so this is one of those things that we sometimes refer to as a superbug because it's hard to treat. So it's picked up all kinds of genes that help it to be resistant to antibiotics. So we also look at the characteristic of the cell wall, and we can determine what kind of cell wall bacteria have by doing a Gram stain. So Gram stain is differential. You can get either purple or pink as indicators in the cell wall. If you have a gram-positive bacteria, then that's going to indicate that the bacteria has a thick cell wall, and you'll be able to tell that because they're going to look purple. If you have a gram-negative bacteria, it's going to have the thinner peptidoglycan cell wall, and it's going to be either pink or red. So in this photograph, you can see it's a culture that's mixed, and so there's a couple of different bacteria there, and some of them are gram-negative, and some of them are gram-positive. Okay, so we looked at this picture already, but I do want to mention, if you're looking at the cell wall type on the right-hand side, that would be the thin cell wall, and we're talking thin in terms of the amount of peptidoglycan. But many times, not all the time, it's not a general rule, but many times bacteria that have the thinner cell wall that stain gram-negative, so they stain red, are more likely to be resistant to penicillin-type antibiotics. And so when they look at whatever kind of bacteria you have an infection from, they're going to determine what kind of antibiotic to use, partly by what kind of cell wall the bacteria has. The bacteria on the left with the thicker cell wall that are gram-positive, they're a little bit more susceptible to penicillin-type antibiotics.